I would like to introduce you to the systems that are provided by Hugo Sachs Electronics. And in the third step, I will talk about our components from multi-channel systems, namely the flexible MEA, the portable ME system. And then in the next part of my presentation, I'm going to talk about the software. And in the last part, I'm going to show you some data that we recorded in various labs. Some data you will see are recorded at the Natural Medical Sciences Institute here in Reutlingen, but you also will be able to see data from a famous lab in Israel, from the lab of Leo Gepstein, where we did some experiments with this technology. As mentioned, I would like to begin with the setup for the Langendorf system. And I think one of the keywords really is here that it is a retrograde perfusion. So what we are doing here is you have the perfusion inserted into the aorta. And by creating a certain pressure into the aorta, you're closing the aortic valve and you're sending your perfusate solution through the coronary system of the heart. So basically, all the cells in the heart, all the muscle cells in the heart are perfused with your solution. They get their oxygen, they get their glucose, so you can keep the heart alive for a very, very long time. One classical sensor here is the balloon catheter that is measuring the force and the contractility. And of course, we will also look at electrophysiological parameters. And just to give you an idea what kind of products are available from Hugo Sachs Electronics, mostly it is a difference in the size of the animal. So we are going to talk today mostly about small rodents, so about mouse, rat, and guinea pig. But of course, there are also experiments where you work with rabbit, or where you even work with smaller pigs. That can be neonatal pigs, or that can be the classical Göttingen mini pigs. And you see one system that is shown here for all the smaller rodents. That is this small rodent setup. And you will see this setup here in one of the next slides. And you see it consists out of a number of different components. So you have an electronic component, you have a water bath for the perfusion, you have your pumps, you have your monitor with the analysis settings on it. And we are going to focus on this part of the system, and that's where we are going to zoom in on the next slide. And here you see now a heart of a rat. And you can see here pretty well what we are going to do here. So you have here the cannula, a metal cannula that is inserted into the aorta. Here you see the connective tissue of the aorta around the cannula. And you have here the insertion of the balloon catheter. You have electrodes for the ECG recording on the surface. And you also have here a stimulation electrode on the surface of the heart. So everything you see here is a standard setup as it is provided by Hugo Sachs Electronics. And I mention again, this is the setup for small rodents by Hugo Sachs. So now let's bring in the components that come from multi-channel systems. Just to give you an orientation again, we see here again the cannula that sits in the aorta. You see here balloon catheter, and you see electrodes on the surface of the heart. And you see here our miniature preamplifier unit connected to a flexible microelectrode array with 32 recording electrodes. And we have here a grounding socket that is connected via a long cable and this crocodile clamp to the cannula that is in the aorta. And it is absolutely crucial that you connect all the grounds properly. So the system of Hugo Sachs Electronics has in the electronic component a separate grounding socket. Our components, our MCS components, have a grounding socket. And all these grounds have to be interconnected and connected to one stable ground to make sure that you get a very noise-free recording. So what kind of recording are we doing here? We are doing a similar kind of recording as you know from our in vitro setup. We are doing an extracellular recording. And I will not go too much into detail here, but we are not doing something like patch clamp where you really touch the membrane, where you break the membrane of an individual cell. And we are also not putting a sharp electrode into the cell, but we are basically sensing from near the contracting tissue, near the contracting cells of the myocardial tissue. So in our in vitro setup, we have electrodes embedded into the surface of a glass substrate, and you culture the cells on top. What we are doing here is we transfer the grid of electrodes, the array of electrodes, from a very stiff glass substrate to a flexible polyimide substrate. And that allows you to bring the electrodes to the electrically active tissue instead of culturing the cells on the electrode grid. So to give you an idea of the electrode material as we use it for the in vitro design, as I mentioned, we have here our contact pads on a glass substrate, and from here the connecting lanes go to the center of the chip, and you can continue to follow these lines to the electrodes on a glass substrate. And exactly the same thing happens on a flexible substrate, as you will see later on. And for some recordings, we need tiny electrodes with a diameter of 10 microns up to 30 microns. And to obtain a good signal-to-noise level on these tiny electrodes, it is necessary to work with the high-end material, which allows you to get a large contact area to the electrolytic solution or to the substrate, and at the same time keeps the geometrical dimensions of the electrodes as small as possible. And the material we are using for this purpose is titanium nitrate. And you will see that not all of our flexible arrays feature this material. Depending on the signals, on the requirements, we are sometimes able to work with larger electrodes. And in that case, we are also able to work with a material that allows us to manufacture these chips in a more cost-efficient way. So before I show you that chips, let me go to the hardware that is needed to do such an experiment. And you see we have a number of different options to perform such an experiment. I would like to start here at the bottom of the slide where you see the cursor right now. In any case, the element that is interfacing with the flexible array is the head stage unit. And typically we work with either 36 electrodes, 32 electrodes, or 64 electrodes. So for that purpose, you're using one or two of these 32 channel head stage amplifiers. In these head stage amplifiers, we usually have a 2 up to 10-fold gain. And then from the head stage amplifiers, you're going either to a signal collector, and in the second step, to a separate filter amplifier unit, and then you go into the AD conversion unit or into the computer. We have a second module for the AD conversion. You've seen the old PCI slot-based card there. With this version, you have now USB-based data acquisition modules with 64, 128, up to 256 channels. And I already said that is a classical approach. What we are doing for most of the smaller rodent experiments nowadays is we are using integrated systems with 16 or 32 channels. And these boxes really allow you a very simple setup for the experiment. You have connections here for head stages with 16 or 32 channels, and you simply plug in this connector here into this unit, and you have the filter amplifier and the AD conversion in this box. It's one USB cable from here to the computer, and you're ready to go. So this is a very small modular setup that can be a simple add-on to any kind of an existing Langendorf system. Of course, there is one little gap. How do you bring your array of electrodes to the heart? And for that one, we developed this holding and positioning unit that consists of a pretty heavy stainless steel foot and a very flexible arm and a rod mount here that holds the head stage that allows you completely free positioning of the electrode on the surface of the heart. So when we talk about the flexible MEAs, I already mentioned we have different layouts of those. When I talked about the titanium nitrate electrodes, you can imagine that a small signal from a neuron requires these fancy electrodes, and we require a very soft material. And when you look back about 10 years, that was a product we had here at Multichannel Systems, and it was used mostly for recordings in vivo or from isolated brains, as you see here. And you see it's a very soft material, just a few microns thick, that goes in a very gentle way on the surface of a brain. What we're using for the heart is a different material and a different design. Our working horse for this experiment is the EcoFlex MEA 36. 36 is the number of electrodes, including the reference electrode, so it's 32 recording electrodes plus analog, digital, and reference channels. And that one plugs in directly into the 32-channel headstage unit. Electrodes are arranged in a 6x6 layout on the tip of this one. You see the layout of the electrodes over here, and we have the connecting lanes from all sides approaching those electrodes in order to achieve a pretty high density of electrodes here in this area. As I said, that is a very nice design for rodents. We have customers working with 2 of those arrays on the surface of a mouse heart. It's very easy to use those on the rats of the larger rodents like rats or guinea pigs, where you can even do a local mapping on the atrium or on the ventricle and very nicely see local areas. You will see some examples later on. The larger one you see here in this region is something that is used for in vivo experiments on larger animals. 128 electrodes, it's almost a 2 by 2 centimeter recording area. And you see at the contact side, we have here the option to connect 4 head stages with 32 channels each to cover the 128 channels that are located on this array. I have to say that this one is not used for Langendorf experiments, but it is used for in vivo experiments on control animals and on animals that are modified to have a higher risk for a cardiac infarct, for coronary infarct. So that is something that is very close to clinical research, and we have several of these systems in larger hospitals all over Europe. I would like to show you in a short movie how these experiments are performed and what you can see in a real experiment. You see a familiar setup here for the Hugo Sachs electronics. You see the beating heart, and we position here the array on the beating heart, in this case on the atrium. And again, you see with the silver wire, we perform the grounding on the cannula that sits in the aorta. And you see the material is flexible enough that it is moving with the beating heart without losing the contact. It's just a liquid film that holds it in position. For larger animals, you can sew it on the surface, but for a small animal, that is the setup you need. Now we are zooming in on the filter amplifier unit and on the signal collector, as you can see here. So we could increase the number of channels easily, and you see here the recording of 32 channels. You see one channel is jumping around a little bit, but all the others have a very, very nice signal here in the screen. And as that was probably a little bit too fast to really see the signals, I would like to show you typical recordings from the data done at the Natural Medical Sciences Institute. Thanks again to our colleagues at that institution that they permitted us to use this data here. So what you see here in a recording where we have the electrodes on the ventricle is so again, we have here the electrodes on the ventricle, and of course you have now a huge signal for the ventricular depolarization. Here you have your plateau phase of the ventricular action potential, and you go here into the repolarization phase of the ventricle. What you would call a P wave in the electrocardiogram is the far field potential from the atrium over here. So now let's position the same thing to the atrium. You see here we are pressing on the atrium, and you now see we have our huge rapid depolarization peak from the atrium here, and you see a very short signal. Atrial action potentials are usually just roughly below 100 milliseconds, and then you get a far field potential from the ventricular depolarization followed by the ventricular repolarization. And of course, when we get these data, we have to analyze data. Classically, we had our MC-REC software that allowed you to have a look at these data, detect timestamps, and get nice results of this. What I really like now is the Cardio 2D software that gives you amazing results in real time. So you have here on top of the software window, you have a long-term display for 40 seconds in this case. And you see one electrode that is selected here on the left-hand side, and you see it's beating in a very regular fashion, and you can control if everything is fine here. Then you zoom in into one electrode. You see that is the electrode labeled over here that is selected, and you can double check if you agree with the timestamp that is selected here. And you have the option to define your timestamp either in the minimum of the signal or in the maximum, or at the maximum slope in the most rapid component of the signal. Down here we have a plot where we can either display the latency between the earliest and the latest detection timestamp, or as we know the parameters of our array, you can also convert this here directly into conduction velocity. And on this side you see now a false color plot. Red means early, blue means late. So you see very nicely the propagation of the signal over the array. And if you just count the electrodes here, you see that's a little bit more than the 36 you have seen in the array before. This was done on a 252 recording layout with cultured cardiomyocytes. And you see here this white electrodes, they indicate that on certain electrodes we could not automatically detect a signal. So all the black circles means the signal was detected and analyzed completely automatic. And here on this ones, we need user interference to help the software to get the right timestamps for the signal. So let's now have a quick look at a recording that was indeed done on isolated heart. We are looking here at data from the lab of Lior Gepstein at the Rappaport facility at the Technion in Haifa. And you see basically the same plot as before. So you have again here your long-term recording, and you see this heart is beating in a very regular way. We look again here at one particular electrode, have the detection timestamp, and you have your latency plot here. And let me point out one thing. You see we have here a delay of just about 2 milliseconds for the signal propagation over the entire array. And even though cardiac signals are by far not as rapid as neurons, when you want to do a reliable mapping, it is sometimes a very helpful feature to be able to scale up the sampling rate to something of 20 or 30 kilohertz to really be sure that you detect the right points into the depolarization. And I do not want to talk too much now about this false color map of the signal propagation, because I think when we look at raw data and when we look at movie files from this preparation, it will become more clear. You can see in the title we are looking here at the left atrium, and you see how the signal is propagating over the array. Let me just point out what you see here to avoid confusion. Before, you had a false color plot where red means early and blue means late. What you see here is a plot where we translate the time really in the time axis as we have a movie, and the red means a positive voltage deflection and the blue means a negative voltage deflection. So basically you see a nice kind of linear propagation in the atrium here. If we switch on to the next one, you see what happens when we perform here a cryo ablation. I know cryo ablation sounds very clinical, very fancy. Let me tell you how we did that in real life in a simple basic research lab. Every lab has somehow its liquid nitrogen. What we did is we put a cannula for a while in liquid nitrogen, make it extremely cold, and put it just under the array, destroyed a certain part of the tissue, and then remove it again, put the array on top again. What you already see here is that we have an extremely high density of the isochronous lines here. And I think if you look at the movie after the cryoablation, it will become even more clear what happens here. So you basically see that the signal seems to go around a certain area here in the center of the array. Let's run the same movie again and just focus on the center, and I think you can see very nicely that there is no longer this linear propagation, but it's kind of cycling around the center. And if you know a little bit about the generation of cardiac arrhythmia, these re-entry cycling excitations in certain parts of the heart are usually one of the mechanisms how you can trigger especially something like atrial fibrillation. And you see, I think in this movie very nicely, by destroying the conductivity of parts of the tissue, we can trigger a kind of early indicator of this form of arrhythmia. So the next one is something that is a bit tricky to reproduce. We did a recording on the same preparation. So again, we are talking about the heart of a rat on a Langendorf setup. And you see here we have really one center where the excitation starts, and you see the isochronous lines are closed circles here. So we managed to position the array right on top of the sinoatrial node, and you see very nicely how the signal, that is not the one with the cryoablation, how the signal in equally spaced isochronous lines propagates from the SA node into all directions. So let me come now to a summary to sum up what I just talked about. I think the systems from Hugo Sachs Electronics for small rodents really match very nicely with our, especially the smaller systems for 32 recording electrodes, and the higher resolution electrophysiology mapping that we provide with our technology gets you some additional information. You get information on conduction velocity. You get information on the connexins that are linking the different cardiomyocytes. You get information on the conduction propagation system in the heart and on the integrity of the heart. And I think if you're really working on a Langendorf heart and you're trying to study the mechanisms of arrhythmia and how arrhythmia is generated on a molecular and on a tissue-related level, I think this is really information that is quite relevant. The advantage of our technology, as you have seen, especially if you restrict yourself to the number of about 32 electrodes, is it is a very, very compact, simple, integrated systems. You do not need to be an electrophysiologist to operate this. And when we have an open day at the Natural Medical Sciences Institute, this used to be an experiment where we allow people from the general public to position the array in certain positions. And it is usually an experiment that is amazing people when they see how easy it is to get this mapping. And if general public is amazed by these recordings, when you show it to scientists and you just see, you get these maps where you normally spend days in analysis, you get this in real time with every single heartbeat. I think that really makes your life so much easier. And one strong point really is usually you get your raw data, export that to MATLAB. We have hard and software from one hand, and we are working together with people with a background in engineering, with people who have a background in programming, and of course people from the biological side to provide a solution that works in the hand of a scientist. And as I said before, it works basically from a small mouse up to sheep, goat, and pig, so from rodent up to large animals. And that was my last slide for this afternoon, and I would really like to thank you for your attention, and I'm more than happy to answer any questions that might have arisen during this little presentation. Thank you very much again, and looking forward to a nice and lively discussion.
Thank you, Thomas. Yeah, actually we had a question on how long you would normally record, or how long can you record?
Well, the Langendorf heart itself will stay alive for a long time. And if I say long time, it's hours. It is up to 6 to 8 hours. And you can imagine we as a small company have to be cost conscious. What we did to evaluate that system, we worked with a small CRO next door at the Natural and Medical Sciences Institute. And what they usually did, they did their drug testing during the day for 5-6 hours. And then I got a call in the afternoon and say, your heart is ready for recording. And then I continued recording for another 2 hours. So 5, 6, 7 hours is not a problem at all.
Okay. And would you also stimulate via this Plex MEA? Is it possible to electrically stimulate?
Depends. Technically, definitely. We can connect a stimulation adapter. We can use any of the gold electrodes to deliver a stimulus. However, please imagine that we try to make our recording electrodes very, very small. We are usually looking at 30 or 100 microns in diameter. And to stimulate a whole heart, you need to transfer a lot of charge. So if you want to stimulate via these electrodes, you have to go to long stimulation pulses, you have to go to high voltage levels, and you will create a certain level of artifacts into your preparation. It is possible if you have a very nice site, a very good contact, if you're close to the sinoatrial node, but usually if it's just to control the beating rate of the heart, you do have an integrated stimulating electrode into the Hugo Sachs setup already, and that would be my first choice.
Okay. Thank you. Yeah, I think this was it for questions. So, yeah, in case any more questions arise, here you see Thomas's email address and also his direct phone number. I think you're always happy to answer any questions via either contact way.
Just think of the time difference, please.
Yeah. So thank you for attending today's webinar. I hope you learned something. Thank you, Thomas, for the presentation. As promised, we recorded the webinar and I will send you the link during the next days. Please note that we have an upcoming webinar on the Cardio 2D software. Thomas mentioned it during his presentation. It will take place in 2 weeks. Yeah, and again, you see the general phone number and email address below. Please just give us a call or drop us an email if you have any questions. We will stay online for another 5 minutes, so in case any more questions arise, the Q&A window is still active. Thanks again for joining. Have a nice day. Bye-bye.